Well, today we're going to be talking about what's called cell signaling, that is, how cells receive signals. And these signals can be carried by hormones, growth factors, small molecules, things that are in the extracellular space that will interact with receptors on the cell surface. And these receptors are generally transmembrane proteins. And these transmembrane proteins bind to these extracellular signaling molecules, and then that then results in major changes in how the cell behaves. Now, I want to just say that in a sense, we're still talking about the cell membrane. Remember, the membrane, of course, has 50% protein, and the proteins do all kinds of different things. Some proteins are pumps and channels, and some proteins are involved in holding cells together in tissue. But today we're going to be talking about a specific class of proteins, which are receptors, which bind to signals from the outside of the cell. And in principle, what happens is that this extracellular molecule, hormone, or growth factor, or other small molecule binds to the protein which is on the surface, the cell surface receptor, transmembrane protein, and the signal does not enter the cell. What happens is it binds to the receptor, and that binding initiates a allosteric conformational change that causes an activity to be induced or to be activated on the cytoplasmic side of the cell. So the signal itself never gets in, it's just that it binds to the surface, and then you get this change in structure, which leads to a change of activity on the interior of the cell. That change in activity on the interior of the cell then activates other proteins or other enzymes, which then leads to a kind of a signaling cascade, or what we might call an intracellular communication network, that can change the biochemistry of the cell. Ultimately, and quite often I should say, that these molecules which are carrying the signal will interact with transcription factors that will then make their way into the nucleus and then activate transcription of various genes depending on what the signal is, and then change the behavior of the cell, maybe cell division. Now, there's a number of different cell surface receptor molecules that I want to talk about. And here's some examples that are shown below. So one example is the ligand-gated channel. And there's thousands of these that are on the surface of the cell. And the ligand-gated channels bind to some of the, it could be hormone, or it could be growth factor or other signaling molecule. And then, of course, what we know is the ligand-gated channels then open up and allow ions to flow through and enter the cell, and then they can change the physiology of the cell dramatically, particularly if you talk about calcium, which we saw, and activates a whole series of proteins and enzymes through calmodulin that changes the physiology and the behavior of the cell. So the ligand-gated channels are one type of cell surface receptor. There's thousands of those on the cell surface. The other two that we're going to talk more specifically about today, one is called a receptor protein kinase. There's a few hundred different types of these on cell surfaces, and of course it depends on the cell which ones are present. And you can already maybe guess what happens there is that there's a receptor, binds the protein, and activates a protein kinase on the cytoplasmic side of the membrane, which then leads to all kinds of changes in different protein activities. And we'll look at that. And then there are what are called G protein coupled receptors, and there's hundreds of these on the cell surface. And again, different ones with different signaling molecules, and they'll be present on different cell types. So there's many, many, many different kinds of receptors. But all these receptors bind some molecule on the cell surface and then alter their activity by altering their shape through an allosteric conformational change, and then lead to this communication network on the interior of the cell. Now, what I want to say, and we'll see this later towards the end, is that the cell is continuously receiving hundreds of signals, not just one signal like shown here, where you get one pathway, but there are hundreds, thousands of receptors on the cell surface, and they're receiving hundreds of signals simultaneously. And so all this signaling has to be integrated, and the messages that come out have to be integrated such that the cell responds appropriately. It's extremely, extremely sophisticated and complex. Let's just look at kind of the general scheme of what happens. So here's the general scheme. It's kind of what I said before, general scheme of a simple transduction pathway. And so you have the plasma membrane, you have a receptor protein, which is shown in green. You have the extracellular signaling molecule, which again could be a hormone, growth factor, or some other ligand, binds, causes some change in the structure of the receptor, which activates some activity, and there's a number of different activities that can be associated with the receptor. And then you see what they call extracellular signaling molecules. These are proteins that, by virtue of their contact with each other, get activated or inactivated, eventually leading to the activation or the modification of what are called effector proteins. And here they're showing transcription regulators. You get altered gene expression, cytoskeletal proteins, metabolic enzymes. This is similar to what I showed you when we talked about the calcium channel. And when you had a flow of calcium into the cell, it bound to the calmodulin, which then led to the activation of other proteins, which eventually led to the activation of these transcription regulators, cytoskeletal proteins. But there's much more. There can be translational regulators. There can be factors of the spliceosome that lead to altered splicing. There can be factors that lead to changes in messenger RNA stability. So it gets really intensely complicated, particularly if you're looking at hundreds of simultaneous signals on the cell. So this is kind of a classic, very simple signal transduction pathway. And the reason it's called transduction is that the signal is transduced from the outside of the cell to the interior of the cell. So we'll start by looking at the receptor protein kinases because they're simple, and they're intuitively we can kind of understand it a little bit easier. The receptor protein kinases have a single transmembrane domain. That's one of their characteristics. In the exterior of the cell, there's the ligand binding domain. On the interior of the cell, on the cytoplasmic side, there's what's called the signaling domain. And in these receptor protein kinases, the signaling domain is a protein kinase. And of course, protein kinases are enzymes that phosphorylate other proteins. And we've seen that before. We've seen the phosphorylation of the EIF2, the translation initiation factor turns it off. There is the phosphorylation of the RNA polymerase, which activates it. So the protein kinases phosphorylate proteins and they change their activities. We'll look at that in a moment. But what happens here is you have the growth factor binding to the receptor. And what happens is the receptor dimerizes. The dimerization activity is not shown here, but you see the two individual receptors are bound each to a ligand or a growth factor, and then they come together on the membrane. Because the membrane is fluid, they can move, and they dimerize, and that dimerization activates the protein kinase activity. And what I wrote on the bottom there is phosphorylation of proteins is the major intracellular signaling pathway. It's really the major one, because this signaling domain, which is a kinase, will then phosphorylate another protein, which itself can be a kinase and transfer it to another protein, ultimately, let's say, transferring it to a transcription factor, which then enters the nucleus and activates gene expression. So there are many protein kinases. It is clearly the major signaling mechanism. So there's many protein kinases, thought to be around 500. And what they do is they use ATP and they phosphorylate, or they add a phosphate to proteins, and they do that through the hydroxyl, the OH group of 3 particular amino acids, either serine, tyrosine, or threonine. These are the 3 amino acids. If you look back at the chart, these are 3 amino acids that have OH as part of their R group. And then there are protein phosphatases, which remove phosphate groups. They dephosphorylate. There's phosphorylation and dephosphorylation. And this little figure just simply shows what I've been talking about. You can have the protein, which is shown in blue, which can be inactive, like that could be the RNA polymerase. Then there's a signal, which then allows the protein kinase to be active. And then that protein kinase, and it could be any number of protein kinases, then activates the protein through its phosphorylation. And then the protein could be, the enzymatic activity or the activity of that protein could be turned off by the protein phosphatase. So it's signaling by protein phosphorylation, turning off and turning on, or modifying the activities of the proteins. So let's look at a little bit more at the receptor protein kinases. So here we're going to look just briefly at one particular receptor protein kinase, and that is the epidermal growth factor receptor, the EGF receptor. Epidermal growth factor is a growth factor that stimulates cell division of cells that have the EGF receptor on them. So the binding of EGF activates these receptor protein kinase, ultimately leading to cell division. Now this particular receptor protein kinase is referred to, as you can see on the left-hand side, it's referred to as an RTK, which really means receptor tyrosine kinase. It's giving us a little bit more information about the activity of the protein kinase, which is part of the cytoplasmic side of the receptor. And so the activation of this kinase activates a kinase which will phosphorylate on tyrosine residues. Okay, so in any case, what happens here is it's showing you get the binding of the ligand to the receptor, you get the dimerization. The kinase activity is stimulated by the dimerization. Then it activates these receptor tyrosine kinases, which autophosphorylate the interior domain. So there's lots of phosphates that are added, and then the phosphorylated receptor domain then interacts with a whole series of different proteins. So the addition of the phosphate allows protein 1, protein 2, and protein 3 to bind. And these are referred to as activated intracellular proteins. They also get phosphorylated, and then they're released, and then they activate downstream activities through their ability to phosphorylate other proteins. A little bit more detail is shown on the right-hand side. So you get the growth factor bound, then you get the activation of the kinase domain, and that kinase domain then activates one of those proteins, 1, 2, or 3, shown on the left-hand side. One of those proteins would be a protein called PI3 kinase. PI3 kinase is a protein which is a kinase. That goes on, it leaves the receptor and goes on to activate another kinase called AKT. AKT then goes on to phosphorylate another protein complex called TOR. And then TOR has a number of different activities. Some of them include inhibition of protein degradation and stimulation of protein synthesis. So you get a change in the physiology, the activity of the cell, ultimately cell growth. Now, this is a little bit misleading because what it's showing is the activated RTK, receptor tyrosine kinase, activates the PI3 kinase, activates AKT, activates TOR, but this is just a linear portion of the pathway. The pathway branches out. So the activated PI3 kinase activates many, or phosphorylates many proteins, not just AKT. So you have many things going on. The activated AKT activates many proteins, or phosphorylates many proteins. So it's not just linear like this, it's opening up and expanding the changes in the activities of so many different proteins in the cell. Now, what happens quite often, we see that molecules, receptors, or other components that are involved in activating cell division quite frequently are also involved in cancer. That is, defects in these pathways can lead to cell growth even when the proper signal is not present. So you can imagine, let's say, for example, a mutation in AKT. A mutation in AKT which would cause it to be permanently activated would then be phosphorylating all those downstream targets, and then you would get cell growth even if the growth factor is not binding to the receptor. And that's actually what happens quite often. Many oncogenes are parts of these signal transduction pathways. Now, in particular, what I'm going to tell you about is that in many cancers, or in some cancers, there's defects in the epidermal growth factor receptor. That is, defects in the receptor protein kinase. And one that's been studied quite extensively is a certain type of leukemia. So as it turns out, in a number of leukemias, particularly what are called erythroid leukemias, so these are leukemias of red blood cell precursors, that there's a what's called constitutively active EGF receptor. So the defective EGF receptor is responsible for the erythroid leukemia, and what happens is in this case, there's been a deletion in part of the gene, which deletes the exons which encode the ligand binding domain on the exterior of the cell. And so you have a protein that's made that has no ligand binding domain, but it has all the rest. So it gets itself into the membrane by the single transmembrane domain. But the signaling portion, the signaling domain, which is the kinase, is constantly active. And so that's what I've written there. The kinase is always active. So it's the deletion of those exons change the structure of the protein so that it's constantly phosphorylating its targets, and then you get cell division and you get cancer. So the epidermal growth factor receptor, EGF receptor, is also referred to as the ERB oncogene. And historically what happened was the ERB oncogene was identified as an oncogene in erythroid leukemias. And then later, it was known to induce cancers, and people did all kinds of experiments showing that the introduction of this gene will lead to uninhibited cell division. And then it was only later that the EGF receptor was cloned and sequenced. And when they identified the sequence, they found that it was identical in sequence to the ERB oncogene, except it was missing the amino terminal domains, which were the ligand binding domains. So many cancers involve signaling molecules that lead to cell division. So that's the protein kinase associated receptors. And what I want to talk about now are what are called the G protein coupled receptors, which are more complex, and the pathways get even more complex. So there are hundreds of different what are called G protein coupled receptors. The G protein stands for a guanine nucleotide binding protein that's associated with the receptor, and we'll see that in a minute. But the G protein coupled receptors all have 7 transmembrane domains, and they have a ligand binding domain on the exterior or the external part of the cell. And then, just like the receptor protein kinases, the activation or the signaling domain is on the interior of the cell, but it's a little bit more complicated, and it doesn't activate an enzymatic activity. It activates a allosteric conformational change that allows the binding of proteins to it, which then get activated. Okay, so the first step, so there's a number of different steps that occur upon the activation of a G protein coupled receptor. The first is it interacts with these G proteins. That's why it's called G protein coupled receptor. So let's look at the G proteins. So the first step in activation via a G protein coupled receptor is the binding to the G protein. Again, the internal signaling domain does not activate a enzymatic activity like the receptor protein kinases, but it activates the ability to bind to a G protein. And G proteins are shown here on the right-hand side of that little figure is kind of orange and brown. It's a complex of 3 proteins called alpha, beta, and gamma. The alpha subunit binds to a nucleotide, and the nucleotide participates in allosteric conformational changes in the structure and function of the G protein. So on the right-hand side, I wrote a little bit about what happens. So with the G protein, it binds to the guanine nucleotide. So it's either guanosine diphosphate or guanosine triphosphate. When it's binding to the guanosine diphosphate, the G protein is considered off. It's not functional. Then there can be an exchange. The GDP comes off and the GTP comes on. When it's bound in the GTP state, it's active to do whatever it does. And we'll talk about what it does in a second. So what happens with the G protein coupled receptors? Again, the first step, binding of the signaling molecule or the ligand to the receptor, changes the conformation and allows the affinity to change, and so it binds to the G protein. The G protein then gets activated, again through allosteric conformational change. It releases the GDP and then accepts GTP, and then the GTP bound form is released, and the activated complexes are released from the G protein. These proteins are bound to the interior part of the cell membrane by fatty acid, and that's kind of shown by that little red squiggly line. So they don't leave the membrane like some of these kinases that we'll talk about. They're actually bound to the membrane and through the mobility move around. So these activated G proteins, they don't have a new enzymatic activity. They're activated to interact with other proteins and change their activities. So they're not kinases, but their structure is changed so that they bind to other proteins and activate them. So here's an example. So there's certain G proteins that the way they work is they open ion channels. So this occurs in the heart. So here we have acetylcholine, you have A, B, and C. In the A portion, acetylcholine binds to the G protein, the G protein coupled receptor. That G protein coupled receptor activates the G protein, and they move around in the internal part of the membrane. And in this particular example, the activated G protein contacts a potassium ion channel. And that's shown in B. When it contacts the potassium ion channel, and it only contacts it when it's activated, then it opens the channel and allows the flow of potassium into the cell, which changes the physiology of the heart cell. And then through other pathways which deactivate the G protein, where you have the GTP getting hydrolyzed to GDP, it becomes inactive and the potassium channel is closed. So here we have the binding and then the opening of a potassium channel, which then has huge impact on the cell. And so you can say in a sense that this particular calcium channel is really a ligand-gated channel. And the ligand is the activated G protein complex. That's one pathway, and that occurs in heart cells. But there's another pathway which seems to be much more common, and that is the activation of the G protein leads to activation of an enzyme called adenylate cyclase. Adenylate cyclases create what's called cyclic AMP, which is now another messenger in the cell which can activate other proteins or activities, much like the calcium activated the calmodulin. Let's just take a look at this for a second. So many G protein coupled receptors, they activate adenylate cyclase. And let me just tell you what adenylate cyclase does. So at the top of the diagram, it shows ATP. That's classic standard ATP. It's used in making RNA, also used as an energy source. But the adenylate cyclase cleaves off the last two phosphates and causes it to cycle on itself, so that that first phosphate, that alpha phosphate, makes a new phosphodiester bond with the 3' OH. So you get that cyclical bond, and that's referred to as cyclic AMP. Cyclic AMP is what's referred to as a second messenger, because the cyclic AMP can diffuse to other parts of the cell and then activate other proteins through binding to them. And then there's ways to decyclize this thing. There's called cyclic AMP phosphodiesterase that cleaves the bond. Now, the cyclic AMP is also a protein that's bound to the surface, the inner surface of the membrane, by virtue of a fatty acid tail. So everything is happening at the surface, the inner surface of the cell, but the cyclic AMP can diffuse. So here's an example. Again, I'm telling you, it's a simplified example of what goes on during signal transduction. Why is it simplified? It's simplified, first of all, because there's hundreds of receptors on the surface that are interacting with different molecules and leading to different activities in the cell, and a lot of these are overlapping, and so they're integrating signals. But in this particular example, we're going to talk about the activation of what's called protein kinase A. And the binding of epinephrine, which is a stimulatory molecule, binding of epinephrine to the G protein coupled receptor, which is shown in the top left, ultimately can lead to increases in blood glucose, increases in energy in the blood. So the pathway here is the binding of epinephrine to the receptor changing its conformation, binding to the G protein, activating it. You can see the G protein is bound to the GTP now, so it's active. And then one of its many targets is adenylate cyclase. And so that's shown in brown. And so you have the activation of adenylate cyclase, which causes the production of cyclic AMP, and that's shown as a second messenger on the right-hand side, the cyclic AMP. And what you see here is that from the activation of one molecule of epinephrine on the surface of the cell, you will get the activation of a adenylate cyclase. Actually, you can get multiple adenylate cyclases activated. But what's happening here, and it's shown by the many different cyclic AMPs that are being released, is the signal amplifies because many cyclic AMPs are produced by the activation of the adenylate cyclase, which is an enzyme. So the cyclic AMP is released into the cytoplasm, and then it binds to different proteins. It's an allosteric regulator. That's what it does. It binds to different proteins and activates them, and it's showing one and only one of many targets, which is called protein kinase A. So protein kinase A is inactive until it binds to cyclic AMP. The binding of cyclic AMP then activates the protein kinase A. And then what's shown towards the bottom is that cyclic activated protein kinase A phosphorylates a protein called phosphorylase kinase. I didn't make these names up, okay? They're coming from the studies. So phosphorylase kinase is activated by protein kinase A. You can see on the right-hand side there's a phosphate bound to it. It's now active, and it will activate something else by virtue of being a kinase, and when it activates is glycogen phosphorylase. Glycogen phosphorylase, once active, leads to the breakdown of glycogen and the release of sugar into the blood. So believe me, this is a simple pathway. Why is it simple? Because the cyclic AMP is released, and there's many proteins that can be activated by cyclic AMP, not just protein kinase A. But once protein kinase A is active, it has many targets. That's what I have written out there. Many targets, not just the phosphorylase kinase. So we're only looking at one sliver of what's actually happening in the cell. So here what's shown in this table is just giving you some example of activities that are modified by cyclic AMP in different tissues. So cyclic AMP activates all kinds of things. So it shows up at the top epinephrine, which we just looked at, in the heart, increases heart rate, force of contraction, in skeletal muscle. It causes glycogen breakdown, which then leads to increased sugar. In fat cells, it leads to fat breakdown. And then in adrenal gland, it leads to cortisol secretion. In a sense, if you see what's happening here, the pathways, there's different pathways in different cell types, but they're leading to a unified response. So the glycogen breakdown leads to more sugar. More sugar supports the heart rate increasing. The fat breakdown also energy for more, more ATP. And cortisol secretion also is involved in what they call the fight or flight response. Now, I want to talk about the G protein for a second. The G protein, of course, is activated every time a G protein coupled receptor is activated. And what I had mentioned earlier when I talked about the ERB and the epidermal growth factor receptor was that many signaling molecules that lead to cell division are found often to be defective in cancers because they lead to, as I said, as an inappropriate response. They can get the response without the signal. And mutations in G proteins contribute to many, many, many cancers. So here it says mutations in G proteins, RAS, I'll tell you about that in a second, contributes to 30% of all cancers. Doesn't matter, you got breast cancers, colon cancers, skin cancers. Mutations in RAS occurs in many different kinds of cancers. So on the left-hand side, you have the normal situation. You have RAS, which is you have the G protein. Okay, let me back up and tell you about the RAS. Okay, RAS. RAS was an oncogene that was discovered early on in the study of the molecular biology of cancer. It was referred to as an oncogene. It was found mutated in lots of different cancers. If you clone the gene, you can introduce it into normal cells. It will cause them to be tumor cells, so it's a very powerful oncogene. And a similar thing happened with what happened with the EGF receptor. People knew about RAS, they knew the sequence for a long, long time. And then when people started studying the signal transduction pathways and they started cloning the genes and then sequencing the genes, they found that the G protein was identical to RAS. And so the implication was that defective G proteins contribute to cancer. So what's shown on the left-hand side is, and they're calling it RAS. So inactive RAS is bound to GTP. Normal signal transduction binding to the G protein coupled receptor leads to the activation of RAS by the binding of GTP, and then you get what they say controlled growth, a controlled proliferation, normal activity. But what's found in 30% of cancers is that there's mutations at a very specific amino acid 12, which changes a glutamine to a valine. Simple single amino acid change. And what happens with that amino acid change is that it forces or fixes, or what I have written there, blocks the G protein or the RAS in the GTP bound state. So the mutation blocks the protein in GTP bound state, and it's active, and then it does what it ordinarily would do. Activates adenylate cyclase, activates the potassium channel, and anything that it should be doing in those cells, it's doing even without the signal, and so you get the uncontrolled cell growth, get the proliferation, whatever it is, and it leads to cancer. Now, what I've been talking about earlier is the complexity, and that is because there's many receptors on the cell surface. So what this shows is the activation of integration of signals, like what I've been kind of alluding to. You have multiple receptors on the cell surface, and those signals have to be somehow integrated and combined in order to get the right signal, the right behavior of the cell. So here it's showing two receptors. It's showing the G protein coupled receptor being bound by a signaling molecule. It's showing the protein kinase coupled receptor, or the receptor protein kinase being bound to its signaling molecule, activating its kinase. The G protein coupled receptor is activating the G proteins. And so what you see here is the activation of these kinases and the G proteins leads to this whole sequence of events that takes place activating different kinases, activating different enzymes, activating adenylate cyclase, and all those are feeding into an integrated signal to get changes in transcription, changes in protein synthesis, whatever changes need to be taking place, take place. And the complexity gets enormous, and I'll tell you, we don't understand a lot of it because it's just way too complex. So this shows one where it's extremely complex, but people have worked out pathways. So here we're looking at 1, 2, 3, 4, 5, 6, 7, 8, 9 different receptors on the cell surface. And you can see all these various proteins that are being activated and modified, and eventually they're showing that what's most important is that the signals get transduced into the nucleus. And once in the nucleus, they change the gene expression. So this is a signal transduction pathway. They get complicated. So in reference to cancer, I want you to look at this. I'm going to start out at the left-hand side. I had mentioned that many of these signaling molecules, because they lead to cell division quite often, depending on the cell type, of course, is that mutations in those proteins lead to cancer, and many of them are known oncogenes. So I'm going to go through this and just point some of these out to you. This is a normal signal transduction pathway, but the following are known oncogenes. From the left-hand side, AKT, known oncogene. RAS, known oncogene. RAF, known oncogene. ERK, a known oncogene. JAK, known oncogene. mTOR, known to be involved in many cancers. NF-kappa B, a known oncogene. That's when they're mutated. RAC, PAC, sorry for the names, but that's what they are. Again, RAS, RAF, ERK, known oncogenes. ERK, by the way, is also found in many cancers. Let's see what else here. SARC up at the top on the right-hand side. SARC, another oncogene. It's a protein kinase. Looking to see what else is there. PAC, P38, another one. Okay, so all these proteins in these signal transduction pathways, or many of them, are, when mutated, are involved in cancer. And also, up at the top, where you have the receptors, of course, you can have mutations in those receptors, like the epidermal growth factor receptor, or B. So it gets really complex, and I just want to close with this particular one, this next slide here. So it's really complicated. We don't understand all the interactions. What's shown on the left-hand side here, well, let me start with the right-hand side. What's shown on the right-hand side is a diagram from a computer motherboard, showing all of the connections that have to be occurring correctly in order to get the programs to function correctly. Now, these are things that we've designed, not we, but these are things that people have been able to design. And they're pretty complex. But it's nothing compared to the complexity of the signaling events and the signal transduction pathways and the integration that needs to take place in a cell. So what's shown on the left-hand side is what's called a protein interaction map. And again, believe me when I tell you this is a simple one. And what it's showing is different proteins interacting with other proteins and the connections between them. So if we talk about cancer, let's say, or we talk about any kind of complex disease, you get a mutation in one of these proteins, and the implications are really intense. Now, if you have a computer and you have a program that is kind of looping, you can't turn the program off. It keeps cycling, whatever. You can go to somebody who knows computers. First of all, you can pull the plug on the thing. But you go to somebody who knows computers and who's worked on computers, and they can get in there and they can somehow reprogram it, or they can fix it. Okay, and that's because we understand the arrays. People who made it. When you're talking about cancer, which is what I study, that's why I keep bringing it up. But when you talk about cancer, we have a mutation in a particular protein, and the cell is undergoing this constant loop, this program of cell division, and you have to go in there and figure it out. It's like, good luck. Find out which protein is defective. Find out what that protein is doing. Find out all the different kind of interactions that take place. So in any case, this is the cell signaling, signal transduction pathway. Is very complicated, very interesting. Okay, I think I'll leave it at that, and have a good rest of your day.